Hi everyone, I'm Marloes Peters and I'm a professor in Bioinspired Materials at Newcastle University in the UK. And in this video today I'm going to talk about how we can draw inspiration from nature to develop new materials that mimic nature in order to combat antimicrobial resistance. And antimicrobial resistance is when microorganisms such as bacteria, fungi and parasites, when they evolve and no longer respond to common treatment, making it much harder to actually find effective treatment. Now this is a natural process, so you have antimicrobial resistance evolving over time, or AMR. However, if there's an accelerated development of AMR, which can be for instance due to the presence of antibiotics or other stressors, this becomes a real problem. Now if you want to know more about how this actually works in the lab, we do have another video where you can see what these materials look like in practice. We talk about sensors today, where I'll show how you combine both the thermal sensing with an optical readout, but we also look at electrical sensing because that's something that you can use for in vivo monitoring in the body. Besides that, we're also really interested in drug delivery, and we look at the sustainability aspect overall. So that means, for instance, we use a lot of polymer materials, so plastics are made of polymers. So we want to make sure that the materials that we produce are sustainable. So you can look at things like material selection, but also for instance at recycling materials and seeing whether we can make new materials out of used plastics. So hopefully you're very familiar with the concept of molecular imprinting. Otherwise, for more details, look at this video. But in brief, what it is, we have something that we want to detect, so that's the target. In this case, it would be either bacteria or an antibiotic. We assemble some monomers that bind with it, so we form a pre-polymerization complex, and then we polymerize around it. And then finally, we extract the target at the end, so we end up with porous materials that have high affinity binding sites that match both the target in terms of size and shape, but also in terms of chemical functionality. And the advantage of this is a very versatile approach. So you could use this for something such as, for instance, small compounds. Think of, for instance, antibiotics. But you can even go to like large bacteria or viruses or other macromolecules. So that's very unique. So you can use this approach for detection of a range of different compounds across different length scales. Also, you have almost an infinite number of building blocks that you can use, whereas in nature we're limited to the amino acids that we have. And most of the time you would use some computational modeling approaches in order to screen what monomers or what building blocks are the best to use for detection of your target. There's a lot of reasons why you would want to use polymers over biological materials. So if you have these polymers, they are low cost and you can produce them at scale. And when you do this, because this is synthetic, you would not expect any batch to batch variation, which you can get in biological materials. Now what's really important here is their robustness. So these are cross-linked materials, which means that they're quite stable under extremes of pH and temperature. And then finally, even though we have recombinant antibody technology, the use of molecular imprinting is an entirely animal-free technology. So there are some ethical advantages to this as well. Now, how can we monitor antimicrobial resistance? Well, actually the key to combating antimicrobial resistance is picking up the hotspots where there's contamination. So thinking, for instance, if there would be a high concentration of antibiotics somewhere, we would want to figure out where it is and clean up that area so it doesn't cause AMR. But likewise, we could also monitor bacteria and check when they start to have properties that correspond to this antimicrobial resistance. In order to do this, we need to do on-site measurements. So for instance, if we think of water, and in this image you can really very nicely see how all of these antibiotics and these bacteria, how they go into our waste streams, which are not all very well regulated. Think of your drinking water, there are some contaminants that they screen for, but not everything. So we want to measure something at the time and the place where this actually kind of occurs. Now, wastewater, you can imagine, that has like a higher temperature, also a wide pH range, but even in your normal drinking water, so even in water that you get from the tap, that's lots of other compounds. And ideally you don't just want to monitor on site, you want to monitor for a prolonged period. And this is where traditional sensors based on antibodies can encounter some problems, because these biological recognition elements can't withstand all of the conditions that they have to be exposed for a long period of time. So this enhanced robustness, which is due to the cross-linked nature of the polymers, brings additional benefits in this case. So when I started my PhD, I started working with microparticles. So I developed particles, and here you can see that these MIPs, they have a porous structure. And we found out that as soon as there's some binding in these pores, you block the electrical resistance. Because electricity will always find the easiest way to go through things, and when that path is then blocked, you actually see that the resistance increases. And the electrical resistance, we measure this as with impedance spectroscopy. Now, what is different, what we found out, is that it's not just the electrical resistance. We also thought that we could perhaps transfer this to thermal resistance. So other researchers in my group looked at this and they applied this to other principles. And I found that if I used my microparticles that were designed for neurotransmitters that contain these cavities, if you would apply like a temperature pulse through these materials, then you would see there's a difference between when they're unoccupied, so when there's none of the target present, and as soon as some of these neurotransmitters actually bound to your particles, you can see that the heat flow was blocked, which led to a change in the temperature. And this concept was later patented and was used for using a lot of these molecularly imprinted polymers in combination ranging from cancer cells to proteins, but also for these small molecules. So you can imagine there's a wide variety in these microparticles. So they all have different sizes, and that also means that there's a distribution of the binding affinity. Another big problem is, remember, if you're extracting your target afterwards, and if it's kind of embedded within like a big clunky particle, it takes a long time and it can lead to a problem with diffusion for binding and extraction. Now, what people tend to do nowadays is to move towards either films or nanoparticles. When you use that solid phase, you can separate homogeneous particles with the high affinity that you want. Now, also a big advantage when you work with nanoparticles, they can become soluble in water and they are biocompatible, unlike the big microparticles. So you could start to think of other applications. So what we've done, we looked at a range of compounds, and important for this particular topic, we looked at some antibiotics, including vancomycin, and developed materials for that. In order to make it very simple, what we did is we had some of these polymer materials in a liquid and we functionalized it onto a thermocouple, which is something that measures the temperature. Now, even if they're a nanoparticle format, they still retain that porous structure. So you could see we modified the thermocouple with these materials, and as soon as something bound to it, again your heat flow through your thermocouple was impacted. And we could use that change that was measured by your thermocouple, and bearing in mind thermocouples are really cheap, in order to determine how much of your material was present. So we could quantify the amount of vancomycin that was present somewhere. So vancomycin is not so relevant necessarily for environmental applications, but it's for instance very important in a hospital where you want to know exactly what kind of concentration people have. But here you can see a picture of what this setup looked like. So you can see basically it's a big box. So what's in there is actually a system that can first of all measure the temperature. We want to have something that can control the temperature, which we do with a feedback loop and by having a power source which can imply a certain voltage. And then we also want to make sure that we have Bluetooth connection so we can get our data, in this case in a laptop. And all this was stirred with LabVIEW software. Now the actual sensor is very small. So when you think of water, obviously you're not so limited in terms of the volumes that you're working with. You can work with a big volume, but typically we work with a flow cell which is around 100 microliter. So that's, imagine, the same as 3 drops of something. And then we have a disposable electrode in there, which is called a screen printed electrode. So all these electrodes are only a few pence a piece. Very easy, they're completely disposable. You slot your electrodes in there, you measure, and then for the next measurement you move on to the next electrode. The first measurements we did, we started still working with the microparticles. And the reason for that is these microparticles can really be produced in bulk. So what we did is we had this screen printed electrode, which is a piece of plastic into which we drop casted some of these polymer materials. Now then we started looking at a range of antibiotics. So in this case we looked at amoxicillin, which is very commonly used, not just both for humans but also in the veterinary sector. and we looked at concentrations in the nanomolar range, which is similar as what you would have for environmental contamination. Now here we worked with a molecularly imprinted polymer for amoxicillin, but we also work with a non-imprinted polymer, which serves as a reference. Now a non-imprinted polymer is produced in almost exactly the same way as a molecularly imprinted polymer. What you do is, rather than having your target in the beginning, you just follow exactly the same polymerization process, but without the presence of the target. So in terms of chemical composition, what you get is exactly the same. However, this will not be a porous material that will contain a cavity of binding sites. So all of the binding that you should see to your non-imprinted polymer, that's all non-specific. Similarly, you could also imprint with another target, and that would allow you to test your cross-reactivity. Now, just look at it here. You can see that as soon as we start to increase the concentration of this amoxicillin, you could see there was an increase in the thermal resistance for the molecularly imprinted polymer, which is depicted in red. And the thermal resistance we define as the temperature gradient, which is first of all the temperature that you impose to the system, which is in almost all of the cases 37 degrees, versus the temperature that we measure, that's T2, over the power that you need in order to keep it at a fixed temperature. So under ideal conditions, the temperature that we keep it at and the power are exactly the same, they are fixed. However, if you start to inhibit your heat flow due to binding in your polymer material, we would expect the temperature to go down. So as a result, your thermal resistance will increase. And that's exactly what we see here. And we see there's a gradual step for your molecularly imprinted polymer when you start to increase the concentration. And a spike, that means that every time we did a new addition, because as soon as you introduce some liquid to the system, you will see there's a temporary drop in the temperature due to the addition of a cold liquid. But then, as soon as it stabilizes again, for the nip you almost see a spike, and then it's back to the baseline. But if there's binding to your polymer material, your baseline will end up higher. And that's due to that binding increases that thermal resistance at the solid-liquid interface. And you can use that to convert it into what we call dose response curve, where you look at your response, in this case the thermal resistance, and we look at a percentage increase over the concentration. Now, what I mentioned before, a problem of working with these microparticles is that you have a huge variation in the binding affinity. So these particles are really good at testing classes of compounds. So for instance, you have different classes of antibiotics that all share the same pharmacore. However, within that class of compounds, it's very difficult to get levels of selectivity. So your sensor would pick up on a lot of things, which might also be a problem if we would start to look at real samples in water, for instance. So that's why we decided, and also imagine that these microparticles are not exactly like stable when they're on the polymer surface, they might come off quite easily. We wanted to move towards nanoparticles or towards films that have much better attachment to the electrode and are capable of having better selectivity. Now, if you work in sensors, you know that you need to validate your system. Just one detection technique, most of the time, is just not good enough. Especially when you're working with a system which is relatively novel, so there's not industry that would have heard of this thermal analysis. So you want to have something which is easy to interpret. Something that has, if you have like a standard lab, all the equipment that you need in order to measure is already there. So in that sense, we thought it was interesting to combine this thermal analysis with optical detection, because that's much more accepted industry. And in order to do this, the first thing we had to redesign our flow cell, because we mainly work with 3D printed materials which were not transparent. So in order to do that, we made sure that we could implement a glass slide within a Perspex flow cell. And Perspex, that's fully transparent. So we could have something that we could both place under like a confocal microscope, but we could also plug it into our thermal setup. Then the second change that we made is, rather than using these microparticles, which were definitely, if we looked at prolonged periods of time, were unstable, we started looking at films. So this is a very thin layer that was deposited onto glass chips. And the other difference that we had to make in order to facilitate this optical detection is we had to integrate an optical probe. And initially we looked at fluorescein, which was less suitable because fluorescein unfortunately does very easily respond to environmental changes, and particularly changes in pH. And water can have a very wide range in pH. And then we started looking at anthracene as another optical probe. There's many others that you can use, but we thought that would be a very neat way of combining the both. we worked with a system for tetracycline, which is another antibiotic. Again, a film was developed, a film using 9-vinyl anthracene, and what we did is we exposed that film to concentrations of tetracycline. So as soon as he did that, we could see that there was a drop in the fluorescence. So essentially you saw a quenching, which is because you have binding, and that has an impact on the interaction that the fluorophore can get. So if we similarly just use like a buffered solution, we could see there really wasn't much of a drop. So nothing there that can bind to your polymer material, your optical signal remained more or less the same. When we worked with a reference polymer, so this non-imprinted polymer, and applied tetracycline to it, there was a drop though. But what you can very clearly see here is that that change in the signal due to binding of the polymer was twice as much pronounced for your MIP versus perhaps the non-imprinted polymer, where you might get some non-specific binding to your surface, or also bearing in mind that some of these compounds they might exhibit some fluorescent behavior as well. So while these films had a much better level of selectivity, there was still quite some significant binding to the reference non-imprinted polymer. And you can imagine if we want to measure over prolonged periods of time, so let's say over, for instance, 2 weeks, this can really lead to problems. Because it might be that your whole sensor circle is fouled with other compounds before you can even measure. So that's why we think the way forward is to still look at either nanoparticle systems or by playing around much more with the monomer composition in the films. So by now we've limited ourselves to one specific monomer, which can have like very specific binding with your target, but having either a mixture of these monomers, or to look much more at designing of a supermicromolecular probe, we should be able to embed better levels of selectivity. And what we then hope to do is to have like a chip and at the same time do our optical measurements and the thermal measurements simultaneously and see whether the signals actually match up. The big advantages of working with these nanoparticles is that they are extremely versatile. So there's a type of standard recipe for protein, but it's very straightforward to apply it to other applications. So you could look at things at, for instance, a range of analytical techniques, such as NMR, ITC or computational modeling, in order to adapt your polymers to whatever you want to detect. And bearing in mind for proteins, there are also some standard recipes that you can play with. If you wanted to look at high throughput screenings, there's also ways of how you can use pseudo-ELISA assays to quickly work out which polymer composition works the best. What are some of the conclusions that we can draw from this? Well, we know monitoring is key in order to prevent accelerated development of antimicrobial resistance. So we should have on-site measurements that can either look for contaminants, such as antibiotics, or for changes that might occur in microorganisms. And the advantages of using these polymers is that they are robust. So if we wanted to look at either optical or electrical detection, we can incorporate probe molecules in order to very quickly determine what's happening. And in the future, especially because of the high cost, the limited robustness, and the ethical concerns around antibodies, it would be great if we can replace all of this with biomimetic materials that can actually outperform nature. Thanks for watching. Do have a look at our playlist Bio-Inspired Materials if you want to know more about this topic.